Hey there, lab technicians! Ready to dive into the world of paraffin wax tissue processing? Today, I'm breaking down the principles and practice of this fundamental technique that's crucial for a range of tissue samples. Let's get started. First up, why paraffin wax? This material is fantastic for preserving tissue morphology and allowing us to cut ultra-thin sections for microscopic examination. It's like the unsung hero of histology labs everywhere. So, how do we get started? The first step is fixation. This is where we treat our tissue samples with chemicals like formalin to preserve their structure and prevent degradation. Think of it as hitting the pause button on tissue decay. Next, we move on to dehydration. Tissue samples are soaked in increasing concentrations of ethanol to remove water. Water and paraffin don't mix well, so this step is crucial. It's like prepping for a paraffin party. Now comes clearing. We use a substance like xylene to remove the alcohol from the tissue, making it ready for the paraffin infiltration. This step ensures our samples are completely free of water and alcohol, paving the way for optimal paraffin embedding. The star of the show, paraffin embedding. Here, tissues are infiltrated with molten paraffin wax, then placed into molds to solidify. This process provides a firm medium to support delicate tissue structures during sectioning. It's like giving our samples a comfy wax blanket. Once embedded, it's time for sectioning. Using a microtome, we slice our paraffin-embedded tissues into thin sections, typically 4-6 micrometers thick. These sections are then floated on a warm water bath and picked up on microscope slides. Patience and precision are key here. Finally, we move to staining. Common stains like hematoxylin and eosin bring out the intricate details of our tissue sections, making cellular structures visible under the microscope. It's like adding a splash of color to a black and white photo. And there you have it. Understanding paraffin wax tissue processing isn't just about following steps. It's about knowing why each step is essential. So the next time you're in the lab, remember this guide and process those tissues like a pro. In the laboratory, various types of processor machines are used for tissue processing. These include automated tissue processors, microwave tissue processors, and vacuum infiltration processors. Each type has its own advantages and is chosen based on the specific requirements of the tissue processing protocol. Thanks for tuning in. Keep processing and keep discovering.